So now I'd like to move to introduce our morning's special speaker, George Yancopoulos of Regeneron. George received his MD and PhD in biochemistry and molecular biophysics from Columbia University here in New York. He's a member of the National Academy of Sciences and the American Academy of Sciences. At Columbia, originally he worked with the legendary geneticist Dr. Fred Alt as his first graduate student in molecular immunology, which was probably a bit ahead of its time at that time, and worked with him for almost 10 years before becoming an assistant professor at Columbia. Then in 1989, with Dr. Len Schleifer, he began the company Regeneron, and actually Len convinced George to join Regeneron only after Len underwent an interview from George's father. And George then joined Regeneron as the chief scientific founder and is now the chief scientific officer and president of Regeneron Laboratories. George is a very active scientist, for those of you who haven't followed Regeneron. He's the principal inventor and developer of Regeneron's 4 FDA-approved medicines, as well as of Regeneron's foundational technologies for target and drug development, including, it's a long list, its proprietary trap technology that led to 3 marketed drugs, including Eylea, the blockbuster treatment for various blindness-causing diseases. Velocigene, which is a high-throughput technology for manipulating mouse genetics, as you'll hear more about from George, and Velocimune, the genetically humanized mouse model technology that enables the production of fully human monoclonal antibodies. And together, these Veloci tools have produced Regeneron's pipeline of over a dozen fully human antibodies targeting cholesterol-lowering, rheumatoid arthritis, atopic dermatitis, asthma, pain, cancer, and infectious disease. Regeneron today has a market cap of, I checked this morning, over $39 billion and sales of $4 billion, and has over 4,000 employees. Forbes ranked it as the #3 most innovative company in the world. And if that wasn't enough, in 2014, Dr. Yancopoulos and his team have launched the Regeneron Genetics Center, a new initiative in human genetic research that has already sequenced over 120,000 people. And please welcome George Yancopoulos.
Thank you, Tom, for that nice long introduction.
Yeah, well, thanks for being here. Well, we can actually see Regeneron from here, we've discovered, if you look out that way.
It towers over the city. I'm only kidding. But I really appreciate the opportunity to be here with your esteemed investigators, and also the opportunity to just, what an incredible highlight. I mean, this building and these views. I should mention that, you know, we are in the New York Academy of Sciences. I've been a member of the New York Academy of Sciences since I was 14 years old. I may be the longest standing member of the New York Academy of Sciences, but I don't think I've ever been in this building before, so there we go.
Thank you. Well, we're just going to conduct this in a little bit of a fireside chat mode, or breakfast chat, I guess, mode, and then throw it open to questions and, you know, just continuing discussion with all of you. So first of all, George, congratulations on obviously leading Regeneron to such incredible impact on multiple disease states. You know, with success, for example, in macular degeneration, high cholesterol, sometimes one could expect that a corporation or any organization like Regeneron could focus on what it's good at. Can you tell us a bit about your philosophy or strategy for keeping a broad eye, keeping a broad landscape open?
Well, that is something that we think distinguishes us. At Regeneron, we have a saying that focus is a dirty word. And part of that, if you think about it, which is that, you know, everybody always talks about, you know, thinking outside of the box. You can never think outside of the box if you're focused, you know, in the middle of the box. But in industry, what has been the rule in the past And you guys, most of you guys are not from industry, so you don't maybe know how it works. But there's about 5,000 biotech and biopharma companies in the United States. 5,000. You know how many drugs get approved every year? 5,000 companies, all trying supposedly to come up with a new medicine. How many? 20. Isn't that depressing? I mean, the odds are overwhelmingly against you. Okay, and basically it means that, you know, every year at least, you know, 4,980 companies don't achieve that year, you know, one of their goals. And what all these companies, most of these companies do, especially the smaller companies, they focus. They're built on one idea. It usually comes from academia, and then they try to develop it and focus on turning that one idea into a product or a new medicine. And every once in a while, one of them succeeds, and they say, wow, this is how you do it. See what they did? They took that one idea, they focused on it, and look how great it turned out. But people don't realize that that's not happening in a vacuum. That's not the recipe for success. That's the recipe for success 20 out of 5,000 times. Because, you know, any one idea has such a low chance of actually working. So I'm not sure we totally understood all this early on, but we totally understood, I think, science, because we consider ourselves fundamentally still just basically scientists, like everybody else, just trying to apply it in a slightly more, you know, therapeutic way. So, and we recognize that you don't do science by focusing. So there was enormous early pressure from the outside world, from our board. Every time we came up with one discovery, a lot of our discoveries came either in our own labs or in collaboration with, you know, academics. And every time we had something that looked like a good idea, I can't tell you how much pressure there was. Hey, guys, forget everything else. Just focus. Turn this thing into something. And once this thing is approved and you guys have some revenues, then you can go back, you know, and worry about the science. And luckily, we were able to sort of fight that impulse and those drivers off, because it turns out, I would have to say that for the first 10 years, you know, it took us 20 years to get, you know, our first really successful product. Certainly, any of the ideas that we had in the first 10 years that anybody thought, including me, might turn out to be a winner, did not. So if we had focused on any one of them, well, I wouldn't be here. If I was here, I might be, I don't know, you know, pouring the coffee. So I think that what we learned, and now other people are sort of appreciating, but it's not so easy to actually do, is that if you're going to build an organization, a biopharma venture that really has ability to be bulletproof, that is to continually come up with new potential therapeutics across all biological arenas. It doesn't matter whether it's for asthma and allergy or whether it's for, you know, cholesterol lowering and heart disease or for even brain diseases. You have to have incredible depth in the organization and this ability to think broadly and widely and make connections, because that's what science is all about.
You know, I guess I covered some of these, but can you give us some stories about Regeneron's past, about how exactly keeping an eye on the horizon, so to speak, led to unexpected discoveries?
Yeah, well, I think a lot of it is the story of how commitment to certain kinds of just new technologies. We're big believers that in biology, sometimes you can't even think of questions unless you have a new technology. And that's one thing that we think we're a little bit special in, that we're always parallel developing technologies which enable our own scientists to then think outside of the box in completely different ways, because they now have new toolkits. So, for example, very early on, in the 80s, we had sort of two separate and unrelated sort of ideas at the time. Okay, one of which I had with my mentor Fred Alt was to make a mouse with a humanized immune system. The idea being, if you had a mouse with a humanized immune system, you could challenge it with things that you challenge humans with. If the mouse responded, and you could test 1,000 mice, 999 die, let's say, I don't know, from AIDS, the one that survives did something in its humanized immune system which you could maybe pull out and clone out and turn into an actual cure for humans, because it's human. So that was the idea that we had back in the 80s. And the way we proposed doing it back then was with technologies that were available in the 80s, having to do with transgenic technology and little bits of DNA. And actually 2 companies went ahead and actually did that were moderately successful companies for a time, though they no longer exist, Medarex and Abgenix, and Fred sort of worked with them to do that. But it was using, like I said, the technology that we proposed in the 80s. But that was like one big idea that we had that we were always interested in. In fact, because I was interested in it, we actually collaborated with Medarex for a long time before they ended up getting bought out, because we very much believed in that approach. But those mice didn't really work that well, if anybody was in the field and knew how those mice worked, because they were really immunodeficient and they didn't really have well-functioning human systems. Well, another thing that we were doing, seemingly totally unrelated around the same time, is we very much believed in mouse genetics, really almost in the earliest stages of doing things like knockouts. So it turns out that at Columbia, one of the groups, Liz Robertson with Argus radiatus and a guy named Tom DiChiara, did one of the actual first knockouts. And the guy who did it, the guy who did it in the lab, was actually a friend of mine. And I thought that, well, if we're going to be starting a biotech company, the thing that we should be doing is we should be doing knockouts, because that's the best way to do genetics, to understand a gene and whether a gene could be involved in a biological process or a disease process, and get insight, validate targets. So I remember giving presentations on this notion, to investors, and I was saying the company that can rule the genome, will rule the industry. And they were looking at me like I had 3 heads. They had no idea what I was talking about. But I brought in my friend, Tom DiChiara, who had done that first knockout. And so shortly after the first knockout was done in academia, we were doing them at Regeneron. And we really saw the value from day one. We, discovered a neurotrophic factor, like BDNF or neurotrophin-3. Nobody knew what it did. We knocked it out, and boom. Instead of years and years, I mean, you guys have to understand the era. It was a totally different era. Instead of spending years culturing neurons in a dish and trying to figure out what they did, all of a sudden you saw, holy cow, this mouse, I mean, it had, no proprioception. I mean, basically, it had no idea where its limbs were, and you could immediately, figure out which neurons were involved, and the whole biology became clear. So we saw this power of the mouse genetics, and we started doing it the old-fashioned way. And then we just kept trying to improve the technology, because we were frustrated. At the early days, it would take us, just like everybody else, a postdoc and a technician, a couple of years to do one knockout. And we were saying, wow, at this rate, we're never going to do anything. So we eventually developed this velocigene technology, which even in this day of CRISPR and all these other approaches, I think still stands alone as the most powerful technology. Nobody else can manipulate in one step hundreds of kilobases of the mouse genome. So what we can do with velocigene is we can precisely replace a piece of the mouse genome with the corresponding part of the human genome. And we can do it in 300 kilobase steps at a time to the nucleotide. So we can put in mutations, human mutations, where you don't even know where the gene is, and see if you're modeling the disease. But to get back to your question, so we're developing this great velocigene technology, and we actually always had an interest in a humanized immune system in a mouse, and we're working with Medarex, where we're not really satisfied. It's not really giving us the type of cures that we want. And all of a sudden, it's like putting peanut butter and chocolate together. We said, wow, with this velocigene technology, we can actually do the dream job. We can actually genetically humanize every gene that's required to create a human immune system, which involved 6 megabases. For those of you guys, just to contemplate that. So we have mice now where we've humanized precisely to the nuclei more than 6 megabases of mouse sequences to make these mice to have really a seamlessly operating human immune system, which then allowed us to, and it was not really our long-term plan, to become the leaders in sort of the human antibody field, because now we have the best mouse, we have the best technology. And this mouse, by the way, parenthetically, not only enables our pipeline, but it's brought in just this mouse, not to mention the drugs it's already produced that are approved, but just the mouse has brought in over $5 billion in licensing and collaboration revenues. So that's an example of, you're doing different things, you have all these capabilities, boom, you put them together, and, you create a whole new way to have a pipeline, which, I think when we write the book, we'll say that it was all figured out and planned out in advance, but it was all by accident and luck.
Yeah. Can you talk a little bit about your next plans in terms of what disease states are you looking at as an organization now?
Well, once again, so you guys understand how it works in terms of my world and our world. Things take a lot of time, and I think it's actually important, and they have to. I mean, biology, and I hope you guys have informed, Paul Allen of this. Okay, it's not like
Excellent.
writing an app and making $1 billion and all this. I think biology is a lot more complicated. It takes a lot more time. Okay, and then if you really want to go to human beings, it really requires, a very careful FDA-regulated process. I'm the biggest fan of the FDA. I think we should all be protected from a bunch of Yahoo and cowboys, because most people who claim that the FDA is holding them back are people who are more likely going to hurt people than help people. So it takes a long time to carefully test things in human beings. It takes years. So the things that we're working on now, that I'm excited about now, were actually projects that we started 10 to 15 years ago. But the things that I'm excited about now, well, I shouldn't say that way. How do I say it? The things that are that we see are going to change medicine today are the things that we were excited about 10 to 15 years ago in terms of the fundamental science. The things that I'm excited about now in the labs are possibilities, 10 years down the line. So those are the sort of time frames. So when you say what's the next thing or what we're excited about, we're very excited about this new monoclonal antibody that blocks what are known as interleukin-4 and interleukin-13. These are two pathways that we were instrumental in studying in the mid to late 90s. That's how long ago it was. And unbelievably enough, it was clear to us from genetics and a lot of other studies that these two cytokines or interleukins together might be responsible essentially for all allergic disease. And what we couldn't believe was that nobody else seemed to get it or believe it. When I say nobody else, so in the late 90s, we created, at the time, what we thought was the world's best blocker of these two interleukins. And Len and I went out trying to partner it and make deals with anybody who we were hoping could afford to fund the effort and so forth. And nobody would. And we could not convince any partner. And, there's 5,000 companies that work on all sorts of crap. Nobody saw what was obvious, I thought, based on genetics and biology and so forth. And we flash forward, it was not until we made a major collaboration with Sanofi based on our mouse, and they said, well, you had made a trap against this. We don't believe in traps anymore. So we made an antibody against this pathway. And we put it into the clinic. This shows how hard and how long it takes. We probably put it in the clinic in 2009. And the results from the beginning, were incredible. And you rarely see this in this business, where you have a complete first-in-class game changer that also appears incredibly safe. So basically, from the beginning, but now we're finally at the point where we have gone through phase 1, phase 2, phase 3 testing in our initial allergic disease, but we've also been through phase 3 in a couple other settings, though we're not ready to apply to the FDA. But the first one is atopic dermatitis or eczema or allergic skin disease. We studied the most severe patients. These are people who more than 50% of their body is covered with this horrible, itchy, weepy, crusty rash. It's essentially, if you talk to these people, it's like having poison ivy over 50% of your body all the time. They can't sleep. They can't function. It's the disease that's most highly associated with depression and suicide. It's a miserable life. And it's all based on some sort of allergic continuum. These are the most severely allergic patients. They also suffer from all sorts of other allergies. And there's, unbelievably, there's 1 to 2 million people like this in the United States alone. And actually that's adults. There's even more children. And so in any case, this drug, almost from the first injection, really helps people. If you look at the placebo-controlled phase 3 studies, about 40% of the people go to clear. Okay, and on average, the average improvement in everybody is about 75%. I mean, that's really a miracle drug. And you know, if you, like we do, if you talk to the patients, I mean, this really is life-changing. So the FDA assigned it breakthrough status at priority review, and we're hoping, you know, at the beginning of next year, it'll be approved. But moreover, we've already done our first pivotal study in uncontrolled asthma, allergic asthma, and the results are, I think, as impressive. And we've also done it in chronic sinusitis and nasal polyposis and eosinophilic esophagitis. And in animals anyway, we've also done it with food allergies and all sorts of allergen desensitization. And the remarkable thing is, it really looks like this is the magic bullet. It seems as if allergic disease is due to what used to be called this Th2 pathway, exacerbation of the Th2 pathway. And in so many people, that's just exaggerated. When you block these, you just bring it back into control. And unlike other agents that manipulate the immune system that lead to immunodeficiency, these people, because too much Th2 actually makes you immunodeficient, these people tend to also have more infections, whether it's in their skin or in their lungs and so forth. These people actually have numerically less infections than people who are not treated after they go on this treatment. So this is one thing, for example, that right now we are really excited, because this could really change an entire, you know, disease category that really is impacting so many people and so many lives. But that's what's coming out now, and the science that we did for that, unbelievably, was 10 to 15 years ago. What we're doing now, we're really excited about, but it's not going to have impact, you know, 5, 10, 15 years in the future.
You know, a topic that many people here are working in and interested in is neuroscience, generally speaking, CNS function. And some people may not know the history of the term Regeneron, but it's one of your early personal interests and desires, I guess I'd say, in science, was understanding regeneration. And maybe that's part of the origin of the company name, but in particular to address neural regeneration and repair. So it's kind of a personal science question. Are you still fascinated by the regenerative process in general, and how far are we from, you know, figuring out how to do regeneration in mammals for CNS or any other aspect of mammalian regeneration?
Well, what Tom's referring to, who here knows the old Westinghouse competition? You guys heard of that? Yeah. Okay. Then it became the Intel competition. By the way, do you know what it is now? Yeah. It's the Regeneron Science Talent Search. We just took it over because Intel backed out last year. But and we're very proud of that, because we really think that it is it's a national treasure that can really inspire a lot of young scientists and so forth. But what Tom's referring to, I did a Westinghouse project 40 years ago now, and it was on regeneration. And obviously our company is called Regeneron for regenerating neurons, and that's what we first set out to do. And luckily, we didn't focus on that. but we have continued to work on it. And neurodegenerative diseases in general. And early on, we thought, and we still think, that they can be contributory. I mean, we cloned a lot of the nerve growth factors after NGF that the world, you know, works on and uses today, from BDNF to CNTF to the neurotrophins and so forth and so on. But we have continued to have a really profound interest, particularly in neurodegeneration, how to deal with it and get around it. And I do think that that's an area where there's been, for the first time, some huge breakthroughs. At least I consider them huge breakthroughs. And I think it really, I guess it really probably started, you know, I'm sure everybody's familiar with the prion story. It just seems like these are all perhaps prion-type related diseases. And I think for the first time, that gives us a little bit of a mechanistic understanding. There's still so much to figure out and that we don't understand. But at least I think this gives us, you know, a real handle with which we can start doing things. So we are really making an effort at those sort of aspects of those diseases. In terms of regrowing neurons in certain settings and creating connections and so forth, I mean, we work on that and we still dream about it. But I do think that, regrowing organs or tissues or even cells, certainly cells as complicated as nerve cells, I mean, that's a really long-term horizon.
Let me switch gears a little bit before we throw it open to the audience here, to your personal style. So I remember visiting you at Regeneron a few years ago, or many years ago, I guess I'd say, when we were looking at VEGF and angiopoietins and their combined effect on blood vessel adaptation. And, the atmosphere there, when you presented at Regeneron, I assume it's still true, is really like being at an intensive lab meeting at a university or a research institute. Everyone is sort of bubbling with curiosity and intensity. And you've been described in the press, actually. I was kind of amused to learn as like an assistant professor who's just discovered coffee, which most people can probably already detect. So can you tell us anything about, because one of our commitments is to have all of our investigators always be at the edge. So how do you create this culture in a company that's this mix of curiosity and inquiry? Is it hiring? Is it how you interact with people in the organization? Your goals? I mean, what's the secret ingredient?
Well, it's all of those things. But I have to say that one of the reasons I joined Len to start Regeneron instead of staying in academia, and I hope things have changed a little bit in academia, but I'm afraid, unfortunately, that some of these things have not changed. I actually took a job in my old mentor's Fred Alt's department, and Fred was like a brother to me. And he was 30 years old when he started his lab. I was 20 years old, and I was his first graduate student. We grew up together, and we were best friends, and we were inseparable. And I took a position in his department, and then after I accepted the position, I went out with the departmental chairman. He'll go unnamed, and he says to me, "You realize, of course, that you're not going to be able to keep publishing with Fred now, because you have to prove your independence." And I literally said, "Well, what about Brown and Goldstein, who were two of my heroes, who are now on our board? They were founding board of director members because they were our heroes." But and of course that didn't play well. And they said, "You have to prove that you can do it yourself." And I said, "Why do I have to prove I can do it myself? What if I can't do it myself, but I can do great things with Fred Alt? I mean, does that make, I could come up with a cure for cancer, but because I did it with Fred, it doesn't count?" It's the most ridiculous thing. So there were all these things. I also have to say that even in Fred's lab, Fred was a great guy, and I was his first graduate, but by the time I left, there were like 30 postdocs in the lab, and I could just sense the competition and the fact that I was going in there, and there were people rooting against me. And I hated that. I didn't want that feeling in my stomach. So from day one, we wanted to create a totally different environment. Okay, we wanted to create an environment where people really work together and were incentivized to work together. We have this saying about you get what you select for. Okay, it's true in biology, and we're all a big biology experiment. And so we worked hard, and I have to say, I drew upon everything in my life, like studying every one of my mentors. I like to think that, I pay attention to these things. I played a lot of sports growing up through college. Every coach I saw, team dynamics, all these things. And all I wanted to do was build an organization, I think you saw it, where people are truly working together. But the one difference from a lab meeting that you described, I think you're absolutely right. That's what we strive for. But the difference is the 20 of the people in the room are at the level of like academic full professors. And they're all working together, and they're all arguing, and they're all coming, to a better conclusion together. And what we've done over the years, I really think not everybody can innovate. You got to understand that. A lot of people don't even understand there's all sorts of innovations, there's useful innovations. I've had the opportunity to now select, the people who either can do it, or I think you can teach people, or can be taught how to do it effectively. And I really do think that you can innovate. I don't believe that much in the notion of like one scientist, by himself, brilliant idea. I just see how many of our ideas, I can tell you every idea, from TRAP to Velocigene to Velocimune to, our allergy drug to everything. I was in the meetings when these things happened. It was a group of people brainstorming together, to take something from here, to there that totally changed. And group thought and group think with people who are really able and trained to think innovatively, I think is incredibly powerful and something that we don't do very much, certainly, in the academic biologic world. So I've handpicked all these people, collection of people over the years to form sort of our brain trust. These people, in general, go to every important meeting, and we have what we call like the subject matter experts, with these guys, and these guys may not know anything about the problem, but they know how to think about a problem, and we present our issue, our problem, and then I just let them go. And I think that that's a really, as far as I know, it's a different way from how biology is done elsewhere. I think it's incredibly powerful to have 20 or 30 now minds at the level of full professors engaged, working together, and these people over time have learned, like teamwork, how to think together. And I am really proud of the fact that when we started, we were actually mocked for being so innovative and so scientific. Oh, these guys are so innovative, they're never actually going to come up with anything. They're great at coming up with little technologies and ideas and all that, but they got to focus and turn it into a drug. And now, 25 years later, Forbes, for 5 years in a row, we're among the 5 most innovative companies across all industries. And I think that we still have that spirit and so forth is because that's who we are. And I don't think it's going to change because that's what defines us. That's who Regeneron is. I mean, this really, the whole goal at Regeneron is to think together and to innovate together.
I think, that's a great story about a fantastic culture. We have about 5 minutes or so why don't we turn the conversation open here to the audience? Yeah.
I'd like to hear what George thinks about the comparison between the biotech companies and the big pharma, because there's been a lot of talk and perspective about the failure of pipelines in the big pharma, and they're really feeding off the more innovative biotech companies. Do you see that changing, or are we in a situation where big pharma is really not going to be innovative?
Yeah, well, big pharma right now, I mean, their success simply depends on exactly what you said, their ability to recognize and bring in things from the outside. And it's sort of inevitable, and it's just a horrible model. And it's, so another thing about Regeneron that's different, that speaks right to it, is we are the only company in our industry, biotech or pharma, okay, where the leadership, I mean, the guys who started the company are actually still totally engaged in it, but it's not only Len and me, it's like the whole next layer. I'm proud to say that I've lost one guy, okay, over the course of 25 years that I didn't want to lose, okay? So our people are all lifers. And it doesn't matter whether they came, and we brought them in 5 years ago, or 10 years ago, or 25 years ago. They're not going anywhere. You know why they don't go anywhere? Like Science Magazine just, has said for 4 of the last 5 years, we're the top company to work for in the biopharma industry. Why? Because you get to live your dream. You get to think and innovate, and then watch that become drugs. But you have a whole leadership team, a whole scientific leadership team that's grown up together, that's actually done it. I mean, there's no other company that has one group of people that develop one drug that are all still there. We have our entire team that's developed all of our drugs, and they're all still there. Why and how do we have this? Well, having a leadership team that is committed forever to the company because they feel like it's their company, it's their legacy, is so different. If you're at Pfizer, okay, you know how long you're going to be CEO of Pfizer? Maximum 5 years. And what are you managing? You're managing your P&L. When you're managing your P&L, what does that mean? That means that every year you're worried about pennies of earnings on your share price. What does that mean? Well, anything that's not going to help you this year is not useful. What's not going to help you this year? I just told you that R&D takes 10 to 15 years minimum to be useful to you. So, of course, what do? They cut it out because they don't care what the company's going to be doing in 10 to 15 years. They only care what the company's going to be doing over the next couple of years because there's no long-term interest in management in the venture and in the company. So that's what's happened over time. they say that research is unproductive, and it's probably true the way it's managed, but because it has no positive financial impact for long periods of time, there's no rationale for a 5-year manager to invest in it. So they've all cut R&D. There is no, I can tell you, there's no useful R&D happening in any big pharma company, and it hasn't been happening, and it's dead. And they're totally dependent on the biotech industry. And like I said, the biotech industry has its own problems in that because of the stresses and so forth, people are forced to do this focus thing, which I think detracts from the power that we could have as an industry. So I would love it. I would love it because I think it would be better for the world if there were more people who sort of took this approach that we took, which is sort of a melding of what you do in academia, but in a slightly, turbocharged way, involving, higher level interactions among people towards a therapeutic. If you could do the sort of scientists, the best investigators doing science, but doing it sort of the way, in a company environment, that would be a great contribution. I'm a firm believer in what, academia brings in terms of the basic science discoveries and so forth that we can sort of build on. But in industry, we are doing collectively a lousy, lousy job. And there's very, very few companies that are taking and building on that. And they worry too much about creating value for the next year as opposed to creating value for the next 10 to 15 years. And so it's a sad testament on big pharma, but the industry as a whole.
Ethan? Yeah.
I've understood that that same point that you make, that investors typically want to see a fast turnaround. The big companies, the Pfizer and so forth, the pharma are reasoning that way. How is it that you manage to convince the people who fund you to take that longer vision and go along with you to support a 10, 15-year horizon when others are looking just for effects on the 2-year time scale?
Now, that's a great question, and it's a combination of a couple different things. One is that because we didn't believe in focusing on one thing, what we were continually doing was we were continually generating new discoveries in the labs, that each one of them was being viewed as an independent opportunity. So when we would take a few things and start putting them in the clinic and testing, and those things would fail. So normally, if we had been a focus company, we'd be out of business. But simultaneously, in the background, we had been developing, when we're working on neurotrophic factors, we're cloning orphan receptors, in blood vessels, and we're cloning, angiopotens, and we're understanding fundamental biology about VEGF, so we come up with a VEGF blocker, or we come up with, an interleukin blocker, or we come up with a new technology that other people want to pay for, like a humanized mouse. So what we're doing is, while we're failing in the outside world's eyes, okay, in terms of our clinical efforts, it's not like we're a one-stop shop. It's not like that was it. If that failed, we're out of business, like most companies are. We had these other things, and Len is really, truly gifted master at these deals, because he, this is the other special thing, is that just like I think our science and our view towards research and all that is for the long term, Len's deals were for the long term, and he invented a new kind of deal that now everybody wants, but we were the first to make these deals, and we're one of the few ones that actually make these deals. So the other thing that made our company is that normally when a small company has something that's exciting, and usually, you create value, get it into the clinic, all of a sudden you can sell out. That's what a lot of people want to do. And then they sell out. They basically sell it to a bigger company, and they're left with, maybe they get something that sounds like a lot of money, $100 million up front, okay, for their phase 1 product. But then that's it. They're out of the game, okay? And they're never going to grow as a company. They're never going to, build or anything like that. They're done. They've cashed out. Well, every one of Len's deals was this very unique approach, which was a 50-50 back-end deal. So he said, look, you guys are interested in this technology, this asset, this trap, this ILEA, whatever. First of all, you have to give us an upfront. You have to give us like $100 million upfront to just become players with us. But to be players with us, you have to fund 100% of our R&D. So it doesn't come out of our pocket. So something like 80 to 90% of our R&D is funded by our partners, not by us. Okay? And not only that, so you pay us to get in the game, you pay 100% of the development costs, which is hundreds of millions of dollars, but then on the back end, we're not happy just getting a little royalty or whatever. We're going to be equal partners. We're going to get a 50-50 deal. We're going to get 50% of the profit. So if this really is, if our drug really becomes the cure for allergy or asthma or eye diseases or whatever, it's going to have a meaningful impact on the bottom line for the company. And every one of our deals was like that. And they really worked out. It allowed us to do all this research. So as the research guys were fanning out and doing all sorts of things, everything was being funded by partners, okay? And so it wasn't really affecting our bottom line. When things were failing, we had a million other things going that were continuing sort of the funding and so forth. And when things started hitting, and we started learning, there is a formula to it that, frankly, nobody else knows because nobody else has really done it repeatedly. I mean, you can't, as you guys know, if you have, an equation with a few variables, it's not enough to solve it, right? But if you have enough, equations equal to the number of variables, you can do it. We're the only people who've done it over and over and over again, so we know the formula. So now we're getting a lot of hit rates. So now the whole thing is turning very, very positive in our favor because we're getting more and more hits, and these are 50-50 deals, so we're getting 50% of the back end. Something like EYLEA, it's a $5 billion blockbuster worldwide, and we're getting more than half those revenues directly to our bottom line. So it's those kind of deals that allowed us to fund the research, okay, no matter what was going on with clinical failures and the stock price. So the funding was independent of the stock price, and we did not have to go outside to the markets and sell equity, sell a piece of our company, which is also very special because all the initial founders and insiders, we still actually control the company. We can't be taken over. We can't be overruled by, some outside entity. We control things.
So I think we're almost out of time. Do we have one more quick one? Yeah.
Hi, thanks for that. I have a question about failure. So philosophically, I think most of the people in this room would agree that kind of taking the longer term view means that when you kind of retrospectively look back at it, of course there's a kind of path that that can get through there. So when you're going forward and you have to make decisions about what's not working and when to cut it, I'd just be interested in thinking how you approach that problem, because that's something I think we all have. Failure is something I think we deal with a lot.
Yeah. So failure is an interesting word. A lot of people, labeled us as failures, many times over many years. I like to think that almost nothing that we've ever worked on has failed. Okay, we're just in the process of reinventing the solution and learning from it. So there's actually, and so it goes against the whole grain. I can't tell you how many times, I was advised, by all sorts of people, our board and so forth, to cut things and so forth. I'm not saying if something is a complete dead end, you don't cut it. But if something's not working, and it's an interesting direction, you're trying to cure neurodegenerative disease, you just change your approach, you reinvent it, you think about something else and so forth. So for us, it's a constant evolution. And I like to think that, if you call it failure is just the beginning step to success. And so for us, every single one of our projects, they could have all been stopped at any point. They could have all been judged as failures. But they all ended up, at some point, evolving and being reinvented. And they may end up looking like nothing like the beginning thing, but I can trace every project right now that we're working on to some root that literally it's connected to 20 to 25 years ago. I think that that's how science works. I'm not saying that you can have a hypothesis and it's wrong and all this, but you build on it, you change it, you vary. So the problem, I think, in the industry is that so many things are binary. It's either a 0 or a 1. Okay, it's either a failure or it's a success. And that's how they run companies, because they have one thing, and that thing either succeeds in the clinic or not. Well, if we have something that doesn't succeed in the clinic, we try to figure out why and what went wrong, and then it's the dawn of a new program. So, like I said, I love failure. I mean, I'm not afraid of it. If you know my personal past, I mean, it's filled by what a lot of people would call failures. But to me, I just don't view them that way. And I do think that for people who go into like biotech, and certainly the things that sort of Len and I sort of did, where we skated by many people's views for 20 years, okay, and failing many, many times according to people's views, the team, me and Len and our team around us, those people are, you get what you select for. They're selected, okay, to enjoy the journey, to accept the fact that things are not going to be easy and that you have to reinvent around it. And if you get off on that, okay, then there is no real failure. So I don't know if I answered your question, but that's sort of our attitude.
Thanks.
Thank you.
All right, thank you.
George, this has, thank you for getting us off to such a great start. This should turbocharge our talks for today.